and talk with you about stable reporter flaviviruses and using them for high throughput neutralization testing. So typically, diagnostic ways to diagnose infection are broken down into two different ways. There's direct detection. So you're either isolating the virus and growing it in cells, or you're detecting it by RT-PCR. These are well-established methods, though virus isolation can be tricky. But the drawback is that you can only do these during the viremic period, which can be transient, which leaves us with indirect detection methods. These are typically done by serology and are completed with ELISAs, Luminex assays, or plaque reduction neutralization tests, which are typically considered the gold standard because of their ability to differentiate infections that have a lot of cross-reactivity, as is well documented for flaviviruses. There's a big need for these types of tests, either diagnosing people if they're past that viremic window, for epidemiological investigations where people are asymptomatic or have very mild symptoms, which is a big case for a lot of flavivirus infections. And then definitely if you're evaluating a vaccine, the neutralizing titer is needed to be known for that evaluation. The drawback with the plaque reduction neutralization tests is that they are slow, not amenable to high throughput, and require trained personnel to complete. So the goal of this project was to develop a high throughput neutralization assay where we could still use the high bar that was set by PRNTs, but make it easier and faster to do. So we were going to do this with reporter viruses. Incubate them with a patient serum, infect cells, and then read the reporter's signal instead of the plaques that is typically done. Here's the scheme for a generic flavivirus genome. We have the structural proteins on the left, non-structural proteins on the right, and flanked by the UTRs. One of the best methods for creating reporter flaviviruses is sticking the reporter gene at the 5' end between the UTR and the capsid gene, the beginning of the ORF. To do this, you have to duplicate a portion of the capsid, which is shown here. This is 25 amino acids that are being duplicated, followed by a 2A sequence to separate these gene products, and the luciferase gene, another 2A sequence, and then the rest of the genome, starting with the full capsid sequence. We have in white here denoted this region because we've reduced homology from here to here to try to prevent recombination, which I've demonstrated with this little cartoon here. The polymerase can come off and come back on and completely delete the reporter gene sequence, which is mediated by that homologous region. So in serial passage experiments, as the scheme here shows, where you serially passage the virus on new cells, and then harvest RNA, RT-PCR from the 5' UTR to the bottom of the capsid, we get results that look something similar to this, where these passages, is done in parallel in 2 series. The passages start out at the correct size, as you would think would be there if the reporter gene was there. And then eventually you get a decrease in that size, indicating that there's been a loss of some genetic material. And if you sequence it in this case, you get wild type virus out. To get around this and stabilize the virus, the first thing we did, method number 1, was recombination dependent lethal mutations, where we engineered mutations in this C25 region that didn't block the replication signals that are needed, which is why we have to duplicate this region. But if recombination happened and they were brought within the full capsid, this led to no viral particle formation. We did this both with Zika virus and with yellow fever virus. If you mutate the positive amino acids that are present at the beginning of the capsid and either change the charge completely or just get rid of the charge to a neutral charge, then you can block viral particle formation if that sequence is brought into the full capsid by recombination. So with these stabilizing mutations present, here for Zika and then also for yellow fever, we get consistent band sizes when you do RT-PCR on passaged viral RNA, RNA from passaged virus. This was method number one. It worked very well for Zika and yellow fever. Method number two has to deal with the length of that capsid duplication. As with method number one, if you do 25 amino acids for Zika and yellow fever, you have unstable reporter virus. You get a change in the RT-PCR band size, P4, P5, P6, somewhere around there, depending on which series and which virus you're looking at. If you increase that duplication size to 38, as shown here, for both Zika and yellow fever, you get very consistent band sizes out to P10, indicating that we've stabilized the virus significantly versus its C25 counterpart. And this is also true for all the viruses shown here: Zika, yellow fever, Japanese encephalitis, West Nile virus, which is reported slightly differently, but these last two bands here are the P10 bands compared to the P0 here, and then all four serotypes of dengue virus. So this was a great achievement for our lab, showing that we can stabilize these nanoluciferase reporter viruses so that they can be grown for a continued amount of time, and they don't have to be continually re-rescued from in vitro transcribed RNAs. The goal of this project was to use them in neutralization tests. And not only did we do them in high throughput 96 well neutralization tests, but we also did them in a rapid format. You can seed vero cells the day before infection, mix virus and antibody for an hour, incubation at 37 degrees. And you plate them on those cells, and in as little as 4 hours, you can read those plates and get neutralizing antibody titers because of this stable signal from the bright nanoluciferase. Results using these stable capsid mutations, Zika and yellow fever, are shown here against a panel of sera. This is mouse sera raised against the indicated virus. We get the highest neutralizing titer from homologous sera, shown here and here. And then against that same panel, but against all of the other viruses that we have, we also have results where we do see some, definitely some cross neutralization, as is typical with flaviviruses, but the highest neutralizing titer corresponds with the homologous virus. This is a great first step in showing that these viruses can be used in neutralization tests, but it's a far cry from the clinical data that we would like to see. We did have the chance to test a small amount of human patient Zika positive sera. Now this was against our capsid mutant Zika, and we did parallel. We did the neutralization assay using our luciferase Zika and then a regular plaque reduction neutralization test and compared the results. The correlation is seen here on the right side, and the data is very promising. We also show that these viruses can be used in antiviral testing. This is not a rapid test. 48 hours is still more rapid than you would by plaque, typically by the traditional plaque assay. But 48 hours is given for a significant time for the compound to affect viral replication. We used NITD-008 as a test compound against both the yellow fever and Zika with capsid mutations, and then against our panel with the lengthened capsid duplication viruses as well, and got quite consistent results with what's been reported earlier in the literature. So in conclusion, we have luciferase reported flaviviruses that were stable up to 10 passages. We did this with Zika, yellow fever, Japanese encephalitis, dengue, 1 through 4, and West Nile viruses. And these viruses can all be used in high throughput, rapid, 4-hour neutralization tests, which is a significant decrease in the time, whereas the normal PRNTs can take 3, 4, 5 days to complete. Future directions, we're working with the CDC right now to obtain well-characterized human patient sera that we can validate these viruses in our assay against, so that these can move into clinical use. We're also working on testing these stabilization methods with other fluorescent reporter genes, and then looking at the possibility of having a couple of different colors so that we can multiplex and possibly have a couple of different serotypes of dengue that we can simultaneously get neutralizing antibody titers from. With that, just a couple of acknowledgements. The Xi lab, especially Dr. Shuping Xie, who was instrumental in this work getting started and continuing it. Dr. Fink was the one who provided us with mouse sera. Dr. Weaver has been instrumental in keeping this project going as well. And then our funding.
Dr. Wilson. All right, and now we'll open the floor up for questions for our last 3 or 4 speakers. Dr. Roundy, did we have any questions come in?
Yes, thank you for the presentations, everyone. We did have one question from Dr. Chavez. This is a comment and a question for Dr. Myles. It says, from what you mentioned, it seems using land surface temperatures from satellite imagery might give fine-grained spatial predictions of areas where mosquito infection may be more likely after the cold season, unlike the 8 or 10 weather stations in Harris County might allow to predict. When the sensors were deployed, what were the above ground and below ground conditions? He's thinking about traps that get deployed attached to manhole covers.
I mean, I'm not sure I completely understand the question, but I'll do my best. Yeah, some of the traps were actually deployed in the storm drains, and so the sensors were deployed in those same storm drains. And then other cases, the sensors were just deployed in, various micro climate habitats that were near trap sites. So these would just be, like, as I said, in kind of shaded environments or hedges or any other kind of microclimate features where we thought that mosquitoes might be resting. But I think the satellite imagery is an excellent idea. This was just kind of our initial testing of this, and we were already working with Harris County Mosquito Control, and it made sense to put traps or sensors into those trap sites where they were already collecting mosquitoes, and we already had access to that data. But certainly we'd like to, in the future, expand this much beyond Harris County. So.
Looks like we have a hand up from Dr. Hamer.
Yeah, thanks, Chris. So this would be for Dr. Baker on that on the Arbovirus serology talk. So obviously, yeah, you show a lot of appealing assays for speeding up that more traditional plaque reduction neutralization test, getting it down from multiple days down to a single day or 4 hours. So I'm wondering if you could just discuss for people that would consider using that tool in either a research or a kind of a clinical environment for public health. There are obviously a lot of ELISAs currently being used for dengue or Zika or West Nile. So what could you maybe, the ELISA might take about a day or whatever, depending on the kit and the incubations. So I'm wondering if you could explain maybe the pros and cons of like an ELISA-based approach versus that labeled virus, the rapid neutralization test.
Yeah, I will speak to what I know. I definitely don't know all of the ELISAs. The way I understand it is that they are typically used as a first line test. That's that the ELISA is either like an IgM NS1. ELISA is usually a first line, what's first used to try to diagnose infection. But those can often come back inconclusive based on if there are multiple flaviviruses circulating, whether they've had previous infection with a different strain of dengue or something like that. And then so the next line is then to send it to a reference laboratory where they do PRNT assays to try to differentiate what the actual infection is. And so that would be where this test would find most of its benefit is in this type of a situation, that, and as I spoke to before, in vaccine evaluation or epidemiological surveys. So I don't know if that entirely answers your question.
Yeah, that helps.
But that's at least a start to it.
For sure, yeah. And then these labeled, is it what I think luciferase was the label for these viruses? Is there an intent that those would be eventually available or maybe currently available in that in your UTMB's World Reference Center? Like, is that something that would ever be provided to clients or people interested?
I think if Dr. Shi is on, that might be something that he would need to answer to.
Well, yes, I will speak up for, we usually deposit all the reagents to the World Reference Center at UTMB. We love to distribute it as soon as possible. Really want this to be used.
Mm-hmm.
So yes, we're happy to for this to be used however it can be.
We had another.
By way of a quick follow-up to that question, the Reference Center also has a lot of BSL-2 surrogates for BSL-3 arboviruses that you can use in a traditional neutralization test. So if you're interested in any of those, you can talk to me.
That's a great note, Dr. Weaver. I should note that the Japanese encephalitis and yellow fever virus strains that I used are the vaccine strains, so they're BSL-2 compatible. The West Nile virus didn't have quite as much data on it because it's still in the 3.
Yeah, unfortunately, I don't think there's any BSL-2 West Nile virus specific strain, but apparently the CDC, BMBL, the next edition is going to downgrade West Nile to BSL-2. That's what we're hearing.
We had a question in the chat from Dr. Pei-Yong Shi. His question is for Dr. Myles. He asks, what is the mechanism of the temperature-dependent RNA silencing?
We don't actually know what the mechanism is. We do know that it occurs at a post-Dicer 2 step. So we've done experiments, specifically looking at whether there's a temperature-dependent effect on Dicer 2, and we don't see that Dicer 2 is functioning efficiently. So we actually see in the mosquitoes that are reared at the lower temperatures, we actually see increased production of siRNAs from the viral double-stranded RNA. But obviously that, those more numerous siRNAs are no longer effective at controlling the replication of the virus. So whatever the step is, it occurs downstream of Dicer. And we also know that it's a reversible process, whatever is happening. So basically, the RNAi pathway does recover. If you remove the mosquitoes from the lower temperature and you place them back at the standard temperature, you will see that the RNAi pathway will begin functioning efficiently again. But at this point, that's all we know about it. We have some mutants now, and so we have some additional experiments planned with some of our mutant lines that may reveal some additional details about the molecular mechanisms, but we haven't completed those yet. Thank you for the question.
Okay, yeah, thank you for the excellent questions today, everyone. I'm not seeing any other raised hands or questions in the chat, so I'll push it back to you, Caroline.
Yes, sure. Thank you to all of our speakers today. This was a great way to kick off our meeting, and we look forward to you joining us again tomorrow. With that being said, I'd like to hand it back over to Dr. Weaver quickly for closing remarks.
Thank you, Carolyn. I just want to add, again, my thanks to all the speakers. We had a lot of really excellent presentations today. I've been watching the comments come across, very positive. I think that despite the COVID situation, a lot of projects have made good progress. And tomorrow we're going to be focusing a little bit more on the public health side of our program, which is really more important, if not equally important, to the more translational product-oriented science that you've heard about today. So I really want to encourage everybody to join tomorrow, and especially toward the end of the session tomorrow. We'll learn more about, hopefully, about the future of the program. And we'll have a chance to have some open discussion about what we can be doing to improve the interactions between our academic and public health partners. That's really the main goal of the whole program. So please join us tomorrow, and thanks again for coming today.